Today, we're diving into something that sounds super complicated, but it's actually one of the most elegant and beautiful processes in all of biology. It's called the chemiosmotic hypothesis. And really, it's the story of the microscopic power plant inside every single one of your cells that's generating energy for, well, literally everything you do. I mean, have you ever really, truly stopped to think about it? You eat some food, right? How does that sandwich or that salad become the energy that lets you think a thought, or lift a weight, or even just breathe? The answer is this absolutely amazing piece of molecular engineering. For a long, long time, this was a massive puzzle for scientists. They knew that our cells were converting food into energy, but they had absolutely no idea how. It was a huge energy mystery at the very heart of life. So let's get into it. So here's what they knew. There's a process called the electron transport chain, which is kind of like the engine. It breaks down molecules from our food and releases this big burst of energy. And they also knew that the cell stores that energy in a special molecule called ATP. You can think of ATP as the cell's tiny, rechargeable battery. But what connected the two? How did the energy from that engine get over to the battery to charge it up? It was a complete mystery, a total black box. The solution, when it finally came, was so out there, so simple and radical, that a lot of people just didn't believe it at first. It wasn't some complicated new chemical they'd missed. Nope, it turned out to be a principle of pure physics, just like a dam generating electricity. And this is the guy who cracked the case, a British biochemist named Peter Mitchell. Back in 1961, he proposed this revolutionary idea he called the chemiosmotic hypothesis. It was so groundbreaking, so different from what everyone else thought, that it eventually won him the Nobel Prize. So here's the core of his idea, the absolute key. Mitchell said, forget about some complex chemical messenger. The cell is doing something much simpler. It's using the energy from that engine to just pump protons, which are really just hydrogen ions, from one side of a membrane to the other. That's it. This builds up a huge amount of pressure, creating this powerful gradient. It's pure stored potential energy. The best way to picture it is to think of a hydroelectric dam. You've got all this water building up behind a giant wall, creating immense pressure. That's potential energy. Then you open a gate. The water rushes through a turbine, and the sheer force of that flow spins the turbine to generate electricity. Well, in the cell, the huge buildup of protons is the water reservoir, the membrane is the dam, and a special little enzyme is the turbine. Now, having a brilliant idea is one thing, but science, as you know, demands proof. And because Mitchell's idea was so different, it was met with a ton of skepticism. People needed to see some cold, hard, experimental evidence. So, the first big breakthrough that started to turn the tide came from a scientist named André Jagendorf. He designed an absolutely ingenious experiment to test this whole dam theory, and he used the little green energy factories inside plant cells, the chloroplasts. His method was just beautiful in its simplicity. Okay, so first, he took these tiny sacs from inside the chloroplasts and put them in the dark, so sunlight couldn't interfere. Then he soaked them in an acidic solution, which is just full of protons, and let them fill right up. Step 3, he quickly plopped them into an alkaline solution, which has very few protons. And just like that, he had created a completely artificial proton dam. Tons of protons were trapped inside, with almost none on the outside. And the result? Well, the second he added the raw materials for ATP, those little sacs started churning it out like crazy. There was no light, no electron transport chain, nothing else. Just the raw physical force of protons rushing out was enough to power the creation of ATP. It was absolutely stunning proof for Mitchell's hypothesis. Jagendorf's experiment was a game changer, but it still used a fairly complex part from a plant cell. So the ultimate test was still out there. Could you build this system from scratch? With just the bare bones essential parts? That's where a scientist named Ephraim Rucker comes into the story. What Rucker did was basically build a microscopic power plant from scratch. He started with a simple, man-made bubble with a membrane. Then he stuck in just two things. First, a proton pump that he borrowed from a bacterium, one that gets turned on by light. And second, he put in that turbine enzyme, the one we call ATP synthase. And that was it. So he flips on the light. The bacterial pump immediately starts pushing protons into the bubble, building the gradient. And just like Mitchell predicted, the ATP synthase turbine kicks into gear and starts making ATP. This was it. This was the final confirmation.
It proved that those three simple things, a membrane, a way to make a proton gradient, and the ATP synthase turbine are all that life needs to make energy.
Okay, so the hypothesis was proven beyond a shadow of a doubt. Now let's take a quick look at how this incredible machinery actually works in the real world, inside the powerhouses of our own cells. This whole process is so fundamental to life that it's used in two of the most important energy pathways on the planet: inside chloroplasts, where plants capture energy from the sun, and inside our own mitochondria, where we extract energy from the food we eat. In photosynthesis, everything kicks off with sunlight. When light hits a plant cell, that energy is used to power the machinery that pumps protons into an inner compartment, building up that all-important gradient, that proton dam. Now, in our cells, inside our mitochondria, the process is amazingly similar. The only real difference is the initial energy source. Instead of sunlight, we use the chemical energy released from breaking down the sugars and fats in our food. That's what powers the pumps to push protons out and build the gradient. And in both plants and animals, the grand finale stars this incredible little machine, ATP synthase. This thing isn't just an enzyme. It's a true molecular motor. As those protons rush back through it, they force a central part of it to physically spin. We're talking up to 100 times a second. This spinning motion literally forces the raw materials ADP and a phosphate group together to create that high-energy battery molecule, ATP. So you see, this is way more than just some obscure process in a biology textbook. Understanding chemiosmosis is absolutely critical because it is at the very heart of life, of health, and of disease. It's how you get almost all the ATP for everything your body does. It's even part of how you stay warm. Some of your cells can intentionally leak the proton gradient just to release its energy as heat. When this system breaks, it leads to devastating diseases like mitochondrial disorders. And it's also a target for poisons. A poison like cyanide is so deadly because it instantly shuts this whole process down, cutting off a cell's energy supply at the source. The discovery of chemiosmosis really showed us that life operates on principles of physics that are both incredibly simple and profoundly powerful. It just makes you wonder, what other beautiful, elegant secrets are hidden deep inside our cells, just waiting for us to figure them out?